Welcome to Creating Your Genius.
Artlist.io.
Welcome back to Creating Your Genius. In this episode, I want to do a deeper dive on the mitochondria, starting with its structure and how that's important for its function, getting into the electron transport chain, some essential nutrients that are vital for mitochondrial function, and some of the other benefits they can have for the body, and eventually get into a really interesting group of genes called Sirtuins, which are known as some of the master longevity genes. Going in order, let's start with the structure of the mitochondria. So the mitochondria is an organelle in the cell. The term organelle refers basically to the organs, but at the cellular scale. Just like we have the liver, which processes toxins and builds glycogen, that's the organ in our entire system of the body. But our cells also have these little organs called organelles, and the mitochondria is a very unique organelle because it has origin outside of human and eukaryotic cells. The mitochondria is an ancient bacteria which was enveloped by another organism. When this other organism enveloped the mitochondria, it just so happened that these two organisms were able to live in harmony. We call this a symbiotic relationship, where the organism that enveloped the mitochondria protects the mitochondria, and the cellular processes that the mitochondria were running were able to give energy to this cell. It turned out that this relationship led to better survival for both the mitochondria and the cell that enveloped it, and this relationship continued and eventually evolved into the complex multicellular organisms that are humans, plants, and animals, all which have cells that contain mitochondria in them. Due to the way that the mitochondria was incorporated into this new organism, the mitochondria has what we call a double membrane structure. So it has one membrane that separates it from the rest of the cell, and then within the mitochondria, there's a second membrane which creates compartments in the cell. The innermost compartment is going to be called the matrix. This is where the citric acid cycle takes place, where most of our electron carriers are produced in the form of NADH and FADH2. This is also where the breakdown of fatty acids into energy happens. As we move from the matrix outward, then there is this inner mitochondrial membrane, which is where the electron transport chain is located. And between that membrane and the outer mitochondrial membrane is what we call the intermembrane space. This intermembrane space is going to be very important as we dive deeper into the electron transport chain. Last episode, I mentioned how the electron transport chain is important for how we create ATP, and that it is what the electron carriers NADH and FADH2 donate their electrons to. The entire process of using the electron transport chain to create ATP is what we call oxidative phosphorylation. It has this name because it carries out a phosphorylation process where ADP is phosphorylated to ATP, and the final molecule in the electron transport chain is actually oxygen. When the electrons have progressed all the way through, they are put onto this oxygen molecule and create water. This is part of the reason why we need to breathe to live, is because our cells use this oxygen to complete the reaction that allows us to generate ATP. Taking a step back, the electron transport chain is made up of 4 complexes of proteins and 2 molecules that we call mobile electron carriers. These 6 factors, as well as ATP synthase, are all located on the inner mitochondrial membrane. Each of these factors plays a specific role that allows the electron transport chain to function in coherence. So NADH and FADH2 are the electron carriers that get the electrons from the citric acid cycle and donate them to the starting points of the electron transport chain. And this would be complexes 1 and 2. It's important to note that NADH only donates electrons to complex 1, and FADH2 only donates electrons to complex 2. When I was taking biochemistry, the way we remembered it was FADH2 delivers to 2, and because NADH didn't have a 2, then it would deliver to complex 1. So along with the electrons, the NADH and FADH2 also donate hydrogens to the intermembrane space. The donation of the electrons is actually just the means to get hydrogens from the mitochondrial matrix into the intermembrane space to create something called a gradient. The best description for a gradient is it's like a dam, where on one side of the dam there's a lot of water. There's the dam that's holding that water back from flowing down its normal path. And if the dam is broken, the water is going to rush down without any resistance, and there's going to be a lot of energy associated with the movement of that water. A substance is always going to flow down its concentration gradient, meaning from a place of high concentration to a place of low concentration. This is important for the context of the electron transport chain because to create a gradient, you have to push something against the normal path that it would want to flow. So to think about it, for the mitochondria, you have the matrix, which is going to have a low concentration of hydrogens, and the intermembrane space, which has a high concentration of hydrogens. In order to maintain that gradient, you have to push hydrogens from the matrix to the intermembrane space, and this is going to require energy. The energy that's used to do this is the movement of electrons through the electron transport chain. When the electrons come in contact with each of the complexes, it activates them and they push hydrogens from the matrix into the intermembrane space. The only exception to this is that complex 2 does not push any hydrogens. It only accepts electrons and moves them. So complex 1, when it's activated, for every pair of electrons that it comes in contact with, 4 hydrogens are moved from the matrix to the intermembrane space. Like I said, complex 2, there's none. Complex 3 also moves 4 hydrogens per pair of electrons. And then complex 4 moves 2 hydrogens from the matrix to the intermembrane space, and then 2 hydrogens onto that oxygen to complete the reaction. The mobile electron carriers sort of fill in the gaps between the complexes. These mobile electron carriers are molecules called coenzyme Q and cytochrome C. Coenzyme Q's most important function is to make sure that electrons get from their starting point, so either complex 1 or 2, depending on which electron carrier they get donated from, and make sure they get to 3 without any issues. And then cytochrome C's main function is to make sure that the electrons get from 3 to 4. Our mitochondria do quite a bit of work to establish this hydrogen gradient. So why exactly is it important? The short answer to that would be to create ATP, but that doesn't really give you the whole picture. So ATP synthase, the protein that actually carries out the phosphorylation of ADP, needs some input of energy in order to catalyze the bond and actually get this phosphate group onto ADP. It does this by utilizing the hydrogen gradient established by the electron transport chain. ATP synthase has these little chambers specifically designed for hydrogens to fit in them. And when they open up, the hydrogens, because of the gradient, are going to naturally want to flow into these chambers, because that would be the closest thing to them flowing down their concentration gradient. Once enough of these chambers are filled, the protein ATP synthase actually rotates. And this rotation is what helps the ADP become phosphorylated, ending in us creating a molecule of ATP. The way ATP synthase rotates is very similar to a motor, and ATP synthase has actually been called the world's smallest biological motor, which is, I think, really interesting. So maintaining this hydrogen gradient is essential for proper ATP synthase function, meaning that the electron transport chain has to be always maintained. It's important to make sure we always have enough CoQ10, cytochrome C, and electron carriers in the form of NADH and FADH2 to make sure that we've got enough materials to keep this electron transport chain running. So that's how our mitochondria uses hydrogens and electrons to create ATP, and also why the double membrane structure is so important, because without this intermembrane space, there would be no way to compartmentalize the mitochondria in a way that they could create this hydrogen gradient that can be used by ATP synthase. So we know that we need carbs and fats for energy. And we can use proteins for energy, but that's not the most efficient or ideal source. So we've got the materials, the fats and the carbs, and we have glycolysis in the cell, which we can use to break down carbs. And there's beta oxidation to break down fats, which happens in the mitochondria. So how is this connected? How do we get those fuel sources to the mitochondria so they can be used? Well, glycolysis ends in making the molecule called pyruvate, and we have to get that pyruvate from the cytoplasm of the cell into the mitochondria to be used for energy. And for fatty acids, they have to be transported in their unaltered form to the mitochondrial matrix so they can undergo beta oxidation. In this episode, I'm going to focus just on carbs because each of them have very specific processes and genes and molecules that are important for them being used efficiently. And I feel like it's just the best use of my time to dedicate a whole episode to each one. Last episode, I talked a bit about glycolysis and how it's the main process that our cells use to break down glucose. And at the same time, I talked about how it can be upregulated in detrimental ways when we're dealing with chronic inflammation. With that being said, when there's a healthy cell, a cell that's in balance, there's going to be a balance between glycolysis and oxidative phosphorylation that happens. If glycolysis was never run in a healthy cell, we wouldn't break down our glucose and our blood sugar would stay high and it would end up generating inflammation in the long run. So when glycolysis ends, our glucose is broken down and we're left with pyruvate. We need to get that pyruvate from the cytoplasm to the mitochondrial matrix where it can be incorporated into the citric acid cycle. One of the most important genes for making sure that this process occurs is something called MPC, which stands for mitochondrial pyruvate carrier. Throughout my research, I was not really able to find anything that can stimulate this protein. However, it's been shown that it's very important for making sure that pyruvate gets from the cytosol to the mitochondrial matrix. Another important protein in this process is something that we call PDH. PDH is a part of something called PDC, which is the pyruvate dehydrogenase complex. And PDH stands for pyruvate dehydrogenase. A dehydrogenase is an enzyme that takes hydrogens off of a molecule. This PDH is what allows us to take the pyruvate and break it down into something called acetyl-CoA. And this acetyl-CoA is what is implemented into the citric acid cycle. Pyruvate itself cannot be added to the citric acid cycle. It has to be broken down into acetyl-CoA and then implemented. So this PDH is very important for allowing us to use carbohydrates for energy because it's the link between the pyruvate and the citric acid cycle. So the gene that encodes for a specific part of this PDH enzyme is PDHA1. PDHA1 encodes for the part of PDH that binds to pyruvate. So if PDHA1 isn't working correctly, it may make a version of PDH that is unable to bind pyruvate. When that happens, it's very detrimental to the cell. Because these processes are so tightly regulated, there's another protein encoded for by PDK4, which is the gene, and this PDK4 protein inhibits PDH. So PDK4 is going to become more active when there is an absence of pyruvate, because there's no point in activating PDH if there's no pyruvate to be broken down into acetyl-CoA. So when people do carnivore diets or ketogenic diets, this PDK4 is going to become more activated, and it's going to help switch the body to more fat burning. An important note here is that when pyruvate is either not transported into the mitochondria, or when it's transported but broken down into acetyl-CoA inefficiently, it ends up being converted to something called lactic acid. And this lactic acid is transported to our liver, where it's then used to build new sugars. The generation of lactic acid from pyruvate is also used to regenerate NAD+, actually to take off the hydrogen from NADH so that NAD+ can be reused during glycolysis. And this is part of the reason for some of the fatigue when we're sick, because we need to run glycolysis. So instead of taking that pyruvate and running it through PDH, turning it into acetyl-CoA, and eventually putting it through the citric acid cycle, we're going to take that pyruvate, turn it into lactate, regenerate the NAD+ so that we can run glycolysis. And lactic acid is famous for being one of the reasons why muscles get fatigued and sore after we work out a bunch. A few factors that can lead to an abundance of lactic acid is when there is too much glucose that it can't be broken down into pyruvate quick enough, or if there's a lack of oxygen. These are going to be two of the most important factors that lead to buildup of lactic acid. And the depletion of oxygen is part of the hypothesis as to why lactic acid builds up when we exercise intensely, because as we're pushing our muscles, they consume more and more oxygen, and eventually we can't breathe quickly enough to keep up with that demand. And eventually the stress builds and the body senses that it's in a low energy state. Glycolysis gets upregulated. We need to do more lactic acid to make sure we have the correct factors to allow glycolysis to proceed, and we end up with sore muscles full of lactic acid. Because of the link between pyruvate and lactic acid, PDH has been an interesting target for athletic performance. The idea would be, if you upregulate PDH, you're going to use pyruvate more efficiently, and you're not going to have the potential or as much potential for lactic acid buildup if, as soon as you're making the pyruvate, boom, you're shuttling it right down into the citric acid cycle and you're not allowing it to become lactic acid. The idea is that upregulating PDH may increase athletic performance. PDH's role in athletic performance is illustrated in this study called Effects of Aerobic Training on Pyruvate Dehydrogenase, PDH, and Pyruvate Dehydrogenase Kinase, PDK4, or PDK, in human skeletal muscle. During this study, the subjects trained at 75% of maximum effort 1 day per week for 8 weeks, and their levels of PDH and PDK were tested. There were other genes that were tested, but for the purpose of this, I'm going to focus just on the PDH and PDK. At 8 weeks, both PDH and PDK levels had increased. And this suggests that the body is definitely using carbohydrates for fuel, but at a certain point, it's probably going to switch to fat burning. And the certain point could be when glycogen's so depleted. It could be from fatigue. It could be the fact that maybe the PDH protein just gets used up. But there's a certain point where PDK levels are going to rise in response to exertion. I'm going to take this information and go a little bit of a different way with it. So based on PDH's importance for using carbs for fuel, and it being one of the most important links between glycolysis and the citric acid cycle, I would wonder if exercise intolerance and early fatigue could actually be due to PDH dysfunction. Because if PDH isn't working correctly, there's going to be buildup of lactic acid. And we need energy to carry out everyday functions. We even need energy to sleep correctly. And so if you're unable to be producing energy, unable to be using this pyruvate to create electron carriers and eventually create ATP, it could create a situation where you're tired all the time. And it could create a situation where very little exertion causes extreme exhaustion. And this could be from a few factors. One could be from a PDK enzyme that is upregulated, meaning it's going to be working too well to suppress PDH function. Calcium is a very important cofactor for PDH, and if there's low calcium in the body, it's probably going to impact how PDH runs. Thiamine, or vitamin B1, is another very important cofactor for PDH, and this has actually been shown to increase PDH activity. A pretty fascinating study called The Effect of Thiamine Treatment on the Activity of Pyruvate Dehydrogenase in Relation to Leigh Syndrome. Leigh syndrome is a genetic condition that greatly affects the metabolism of the cells, and it's characterized by symmetrical lesions of the brainstem. In this condition, it's well known that levels of blood lactic acid and pyruvic acid concentrations are high. These markers illustrate that there is difficulty with the body using pyruvate effectively and that it's getting converted into lactic acid. I'm not sure how they discovered this, but thiamine, B1, given in high doses to patients, temporarily improved their clinical presentation as well as helping knock down those high blood lactic acid and pyruvic acid concentrations. The proposed method for this was through thiamine increasing PDH activity. So they took this information and they studied it in rats. And the rats treated with thiamine did in fact have more active PDH complexes, and the levels of thiamine in the mitochondria of these rats was shown to be correlated with the enzyme activity. And the way they tested this was by giving ATP to the rats. In a normal working mitochondria, when ATP is high, it's going to inhibit PDH because there's no need to break down pyruvate into acetyl-CoA to create energy if energy is high. And when these rats were given ATP that had the high concentrations of thiamine, their PDH was not as sensitive to the ATP. And so that's how they correlated the high thiamine being the reason that this PDH became more active. Because if it was a different mechanism, then the ATP should have lowered the PDH levels when there was high thiamine and high ATP in the mitochondria of these rat cells. This isn't to say that thiamine is a cure-all and it's going to absolutely increase your energy levels, but I think it's just important to understand the fact that these vitamins and minerals really play important roles in what's going on in the body. And if you have low energy, or if you want to improve your workout performance, maybe sprinkling in some thiamine might help you achieve your goals. It's all about making bits and pieces of progress and trying to do things that support your body, as opposed to, I'm not saying that thiamine is any sort of cure-all here. It's just that it is an important cofactor for an enzyme that's vital in our ability to process carbohydrates. And based on this, you would have to wonder if the PDH may be downregulated in chronic fatigue, or PDK may be upregulated. And there was a study where serum or plasma blood from chronic fatigue syndrome patients was taken and they exposed cells to this serum, either CFS patient or control. The cells exposed to the CFS serum showed increased lactate production, and this goes back to the point I was talking about earlier with metabolic strain, where for whatever reason, when energy production becomes harder, the body's going to make more lactate to upregulate glycolysis and to make sure that we're able to make enough ATP and energy that we can still function. But just because we can function doesn't mean it's optimal. It makes sense that in a condition like chronic fatigue that there would be something that would be inhibiting PDH and inhibiting our body's ability to potentially use carbohydrates the way that they're supposed to be. And in this study, it shows that PDK, which inhibits PDH, was upregulated and there was increased lactate production. And so based on PDH's importance for energy metabolism and this study with the fatigue and the increased lactate levels, I looked up thiamine and athletic performance, and I found a really interesting study called The Effects of Endurance Training and Thiamine Supplementation on Anti-Fatigue During Exercise. The study said that thiamine intake during exercise positively benefits carbohydrate metabolism in a way that will decrease lactate concentration, ammonia concentration, and anti-fatigue by reducing the RPE. RPE is relative perceived exertion. Therefore, we can consider thiamine intake to be utilized as similar benefits as endurance training. Again, I think this statement shows that thiamine is going to be important for how we utilize carbohydrates. I don't know if you can really go as far as to say that you can cut out endurance training and just give thiamine and get the same benefits to your metabolism. I think the better way to interpret this is that thiamine is something that has the potential to support your metabolism, and it can be an add-on to your workouts. At the end of the day, building the healthy habits is going to be the most important thing. And then you can use this information to make little tweaks to try to help you feel better. I want to switch gears here a little bit and move from pyruvate and thiamine and PDH and go a little bit deeper into coenzyme Q. We often call it CoQ10, coenzyme Q10, or ubiquinol. As I mentioned earlier, this is part of the electron transport chain, and it functions to make sure electrons flow from their entry point, either complex 1 or 2, to complex 3. For the purposes of the electron transport chain, I'm just going to call it coenzyme Q because it switches between the forms ubiquinone and ubiquinol. Before any electrons have been donated, it's going to be in the form ubiquinone, and this is its oxidized form. And when the electrons move through and they reduce it, it's going to be converted into ubiquinol. And after ubiquinol donates the electrons to complex 3, it's going to be reoxidized back into ubiquinone. There are 9 CoQ genes involved in the biosynthesis of CoQ10. We do get coenzyme Q from dietary sources, but as we're growing, most of the coenzyme Q that gets implemented into our mitochondria is actually from what we make. And these genes are important because mutations in these genes have been shown to lead to ubiquinone deficiency. Unfortunately, there's not much research into what we can do to simulate our own body's production of coenzyme Q. However, we can ingest it through diet and supplement with it, but we're not entirely sure if that CoQ10, the form we ingest, actually gets incorporated to the mitochondria. And this is really interesting because although CoQ10 does have antioxidant function in the body, it does seem that its main evolutionary mechanism is as a cofactor for the electron transport chain. And this makes sense because CoQ10 levels are highest in the organs with high rates of metabolism, like the heart, kidney, and liver. The main sources of CoQ10 in our diet come from animal proteins, nuts, and soybeans. CoQ10's antioxidant properties are very interesting because it's been shown to downregulate both IL-1 and TNF-alpha gene expression, and that's the proposed mechanism by which it exerts the antioxidant effect. We're not sure if CoQ10 actually scavenges free radicals on its own, but we do know that it downregulates inflammatory genes. Based on its role in energy-producing reactions in the electron transport chain and its ability to lower oxidative stress, it would make sense to explore CoQ10 in the context of athletes. Supplementation was not shown to have statistical significance for differences in power and anaerobic capacities based on a study called Effects of Acute and 14-Day Coenzyme Q10 Supplementation on Exercise Performance in both trained and untrained individuals. This study is interesting because there was a trend showing increased time to exhaustion in this study, which did mean that the participants were able to exert more. A note for this is that the study tested with 200 milligrams of CoQ10, and it was 100 milligrams twice per day. CoQ10 is safe up to 1200 milligrams, and so it may be possible that for intense exercise, a higher dose might be necessary to generate effects. There's another study testing 300 milligrams of CoQ10 for 6 weeks, and this study is called Ubiquinol Supplementation Enhances Peak Power Production in Trained Athletes, a double-blind placebo-controlled study. And so this was a study done on 100 German Olympic-trained athletes. So these are people that you would think would have very high capacities for power and endurance as a baseline. What it found was the short-term maximum performance did increase. The proposed mechanism could be through increasing the mitochondrial activity by increasing the CoQ10 levels in the muscles for a more continuous ATP production. And this would avoid the ADP buildup, leading to increased glycolysis and decreased mitochondrial function. The proposed mechanism is that the CoQ10 allows ATP to be created more efficiently, and because of that, you avoid running glycolysis too much. Based on these studies and mechanisms, and especially the fact that CoQ10 can downregulate genes like IL-1 and TNF-alpha, this means there probably isn't a diet contraindication for supplementing with CoQ10, meaning that if you're doing keto, carnivore, vegan, there's no diet that you may do worse with CoQ10 on, because it's likely either not going to give you any effects or it's going to help you decrease inflammation. There's one more effect of CoQ10 that's very important, and it links to this group of genes called Sirtuins. Sirtuins are a family of proteins that have wide-ranging but important functions in the body. And like I said at the beginning, they are known for being longevity genes. Their exact function is they take part in maintaining the normal chromatin condensation state in DNA. They're important for DNA damage response, modulating the oxidative stress response, as well as energy metabolism. As our DNA is held in our chromosomes, it's wound tightly into the structure called chromatin. And when genes need to be expressed, it's important that the chromatin relaxes to allow the proteins necessary for gene expression to have access to the DNA. And sirtuins are very important for helping relaxing that coiling, which in turn increases gene expression. Sirtuins help with this coiling via a process called deacetylation. This is the removal of an acetyl group. An acetyl group probably sounds familiar because I've mentioned acetyl-CoA a few times, which is mentioned in the citric acid cycle. It's what PDH breaks down pyruvate into. An acetyl group is made of 2 carbons. One carbon is bonded to 3 hydrogens, and the other is double bonded to an oxygen. And going back to episode 1, acetylation is a post-translational modification, and it's used to modify proteins after they've been synthesized. Let's focus on the energy metabolism part of this equation. So these sirtuins get activated and they go throughout the body and they deacetylate things. So when sirtuins deacetylate a protein called PGC1 alpha, the downstream effects of this leads to control of the mitochondrial gene expression. And these sirtuins there's a few different families of them. Sirtuin 3 or SIRT3 specifically is involved in the process of switching the body into burning fatty acids for fuel and has been shown to be upregulated during prolonged periods of fasting. And SIRT3 is specific to the mitochondria. And one of the mechanisms of fasting and all the health claims surrounding fasting is likely due to an upregulation of these sirtuin genes. Another benefit of SIRT3 activation comes from upregulating the antioxidant response. During oxidative phosphorylation, the mitochondria naturally produces free radicals. It's just a byproduct of running these processes. It's important to have enzymes that can clean up these free radicals, and part of SIRT3's benefits come from coordinating the response of these enzymes. In a study called SIRT3 Opposes Reprogramming of Cancer Cell Metabolism Through HIF1 Alpha Destabilization, it was shown that loss of SIRT3 function was shown to increase free radical production. This finding highlights how important the sirtuins are for coordinating that antioxidant response, especially in the mitochondria, because that's where these free radicals are created during oxidative phosphorylation. Proper SIRT3 function is going to be very important for mitochondrial health. The best way to activate these genes are through caloric restriction and fasting. And like I mentioned earlier, CoQ10 also has the ability to upregulate one of these sirtuins, specifically SIRT1. SIRT1 overexpression in mice helped prevent diabetes. And this study was really interesting. It's called SIRT1 gain of function increases energy efficiency and prevents diabetes in mice. In these SIRT1 overexpressed mice, when they were fed diets that would promote obesity, they still gained weight, but they were protected from the effects of obesity and high blood sugar. And based on that, the study concluded that SIRT1 did make the energy production more efficient, and that efficiency of energy production was protective against insulin resistance and hyperglycemia. The activation of these SIRT1 genes and its importance for blood sugar sort of goes back to how important it is for us to make sure we're not eating too much, and to make sure that when we are eating, we're using our energy efficiently. Because it's a vicious cycle. Things like high blood sugar cause inflammation, which disrupts our energy production, which causes more inflammation, which disrupts our energy production more. And thinking about food and energy production and how much we eat, it's not to say that eating's bad. It's not to say necessarily that fasting is good. It's just that there's a balance in the way that we exist that's very important. And so when you fast, you upregulate these SIRT genes. And when you eat, especially when you eat carbs, you upregulate PDH, which helps you use energy more efficiently. And so there's this balance between all of it. And the way we're able to exist, it's so fascinating because you've got so many different mechanisms going on all at once. And in the same day, you can activate SIRT3 by doing an intermittent fast, and then activate PDH by eating a meal that has carbohydrates. And at the same time, if you broke that fast with just fats, you're likely going to upregulate PDK, downregulate PDH. You're constantly giving these signals to your body, and based on those signals, it's really going to impact how your genes are expressed. And so knowing that fasting upregulates sirtuins, CoQ10 also upregulates sirtuins, thiamine helps upregulate PDH. You can use these little tools to help optimize the way that you're functioning. So maybe you don't want to take B1 first thing in the morning, or if you take a B complex vitamin, maybe you don't want to do it first thing in the morning. so that you keep those sirtuins activated for a little longer. Again, there's no research as to whether B vitamins break your fast or not, but I think one of the things with medicine and the point of research where we're at now is we can look at things and we can look at mechanisms and take it one step further. Knowing that B1 activates PDH, which breaks down carbohydrates, and knowing that breaking down carbohydrates is sort of in opposition to breaking down fats, and it's definitely in opposition to calorie restriction. And so knowing that, you can take that information and take it one step further and say, maybe B1 is going to upregulate genes that aren't really helpful to upregulate if you're not eating. And that approach to the inputs, the genes, and the outcomes that it's going to create, that's what Creating Your Genius is all about. Thank you for tuning in to another episode, and we will be back with more soon.
Artlist.io